Today, we are doing something a little different, and honestly, something I've been looking forward to since we first got our hands on this stack of research.
It's a big one.
It is. Usually when we talk about the human body, we gravitate toward the glamour organs, the heart pumping lifeblood, the brain firing off thoughts, maybe even the lungs.
Right, the big players.
Exactly.
Okay.
But today we are going to talk about the system that actually fuels all of that. And I have to be honest with you, before digging into this material, I thought digestion was pretty straightforward. You eat a sandwich, your stomach churns it up like a washing machine, and eventually, well, nature calls.
The simple version.
But looking at chapter 63 of this physiology text, I was dead wrong.
You are definitely not alone in that assumption. It is, I think, the most common misconception we see. Most people view the gastrointestinal tract as just a tube.
Yeah, just plumbing.
A passive plumbing system. But when you actually peel back the layers, literally and metaphorically, what you find is one of the most sophisticated feats of biological engineering in existence. It is not just a pipe. It is a chemical refinery, an electrical grid, and a command center all rolled into one.
I mean, the first thing that jumped out at me in the intro notes, and this is something we really need to linger on, was this concept of the second brain.
Ah, yes.
I've heard that term thrown around in pop science articles, on health blogs, usually trying to sell me yogurt. But the source material here puts a hard number on it that completely blew my mind.
The number is pretty staggering.
It says the gut has more neurons than the entire spinal cord.
That is correct, and it is a staggering statistic when you really think about it. We are talking about roughly 100 million neurons residing entirely within the walls of your gut.
100 million.
That is not just a few stray nerves connecting back to the brain. That is a standalone, complex nervous system. It is capable of making decisions, managing reflexes, and controlling flow without a single instruction from the brain in your head.
And that is exactly what we are here to unpack today. Our mission for this deep dive is to decode Chapter 63 line by line. We aren't just doing the high school biology overview where we name the organs and move on.
Yeah, we're going deep.
We're going to look at the voltage of the cells, the specific chemical messengers, the intricate plumbing of the blood, and this wild electrical grid that keeps us alive.
Yeah.
We were looking at the general principles of gastrointestinal function.
And to structure this so we don't get lost in the weeds, because believe me, the weeds are very dense and very technical here, we should probably look at this through the four main pillars the text establishes.
Okay, what are they?
We have motility, which is, the movement. Then nervous control, which is that second brain we just mentioned.
Right.
After that, hormonal control, so the chemical managers.
Right.
And finally, blood circulation, which is the supply line for the whole operation.
Perfect. So let's start with the setup, the physiologic anatomy. The text provides this cross-section of the gut wall, specifically Figure 63, Day 2. It describes it as a multi-layered industrial processing plant.
That's a great description.
So walk us through what we are looking at here. If I sliced a section of my intestine and look at it under a microscope, starting from the very outside of the tube, what am I seeing?
Okay, so imagine you are holding a cross-section of a garden hose, but a very complicated living hose. Starting from the very outside, the layer facing your abdominal cavity, you have the serosa.
The serosa, what is that, like a protective skin?
Exactly. It's your protective wrapper. It's thin, it's tough, and it keeps everything contained. But immediately inside that, we hit the muscle layers, and this is where the machinery really begins.
Okay.
First, you have the longitudinal muscle layer.
Longitudinal meaning running the length of the tube, right? Like stripes on a candy cane.
Perfect analogy. The fibers run down the length of the tract. When these fibers contract, they shorten that segment of the gut.
Oh, okay, so it pulls it shorter, like an accordion.
It effectively pulls the tube shorter, yes. Now, move one layer deeper towards the center, and you hit the circular muscle layer.
And these go around it?
These fibers wrap around the gut like rings on a barrel. So when they contract, they constrict the diameter of the tube. They squeeze it.
So one layer shortens the tube, and the other layer squeezes the tube.
Precisely. You've got these two fundamental movements working together. And sandwiched right between those two muscle layers is something we will talk about a lot later, the myenteric nerve plexus. But let's keep moving inward for now.
Got it.
Inside the circular muscle, you have the submucosa. This is a thick connective tissue layer, kind of the structural support.
Okay, holding it all together.
And then finally, the innermost layer, the one actually touching the food, is the mucosa.
And the text mentions something interesting about the mucosa. It's not just a smooth lining like the inside of a cheek. It has its own muscle, right? The muscularis mucosae.
Yes. Sparse bundles of smooth muscle fibers. This is a crucial detail that is so easily overlooked. These little muscles allow the lining itself to fold and ridge up.
Why does that matter? Why does it need to fold?
Surface area. It's all about surface area. If the lining were perfectly smooth, you wouldn't have nearly enough area to absorb all the nutrients you need. By using these tiny muscles to create folds, and then folds on the folds, the gut maximizes its contact with the food. It's an incredible efficiency hack.
It's remarkably detailed, but the real magic isn't just that the muscles exist, it's how they communicate. The text introduces a concept here called the syncytium. I circled that word, syncytium.
It does sound a bit sci-fi, doesn't it?
It sounds like a sci-fi villain organization or a hive mind. What does it actually mean in this context?
Well, hive mind is actually not a bad analogy at all. It refers to how the smooth muscle cells act as a single coordinated unit.
Okay, so how does that work?
An individual smooth muscle fiber is tiny, about 200 to 500 micrometers long and maybe 10 micrometers wide. But they aren't working alone. They are bundled together in groups of about 1,000 parallel fibers.
So they are bundled, but how does that make them a syncytium? How do they act as one? I mean, my bicep has bundled fibers, but it doesn't act like a single unit in that way.
It's all about the connections. This is the key difference. Within each bundle, the muscle fibers are electrically connected to one another through what are called gap junctions.
Gap junctions.
Think of these as open doorways between the cells. They are low-resistance pathways that allow ions, the charged particles that create electricity, to flow freely from one cell to the very next.
So if I start an electrical signal in one single cell, it doesn't just stay there. It's not isolated.
No, absolutely not. It spreads, and it spreads rapidly. If you excite one fiber, the electricity travels to the next and the next and the next. The action potential travels in all directions within that bundle. That is the syncytium effect. The gut doesn't contract in isolated pixels, you know? It contracts in these sweeping, coordinated waves.
That explains so much. It explains why you don't feel a tiny twitch in your stomach. You feel a churn. The whole wall is working together.
Exactly. And the text makes a distinction here, too, which is important. The longitudinal bundles and the circular bundles are mostly separated, but they do have fusion points.
So they can talk to each other.
They can. So usually, if the longitudinal layer gets excited, that signal will jump across to the circular layer too. It ensures the movement is coordinated. If one layer is firing, the other usually knows about it and responds.
Which brings us to the engine driving this movement. Segment 2 of our outline, the electrical battery of the gut. I think this is where the textbook really challenges the average understanding of biology.
I agree.
We usually think of electricity as a brain thing or a heart thing, but the gut has this constant electrical hum.
It does, and to understand it, we have to talk about voltage. The text establishes a baseline called the resting membrane potential. In the smooth muscle of the gut, this averages about negative 56 millivolts.
Okay, negative 56, that's our baseline. That's the ready state. But this voltage isn't a flat line, is it? The charts in the source material figure 63.3 specifically show these constant waves. It's never really resting.
Right. It's always in flux. And there are two major types of electrical phenomena you need to understand here. The first are what the text calls slow waves.
Slow waves. Now, the text is very aggressive in clarifying what these are not. It says in big, bold letters, essentially. These are not action potentials.
And that is the most common misconception. It's the first thing students get wrong. Slow waves are not muscle contractions. They are just undulating changes in the resting potential. The voltage just drifts up and down, usually shifting by about 5 to 15 millivolts.
So if they aren't contractions, what are they? Are they just background noise?
Think of them like a metronome ticking in the background. Or better yet, think of the ocean. The slow waves or the swell of the sea, they just lift the water level up and down rhythmically. They set the potential for a contraction. They determine the rhythm.
The rhythm. Does it change?
Oh, yes. And this rhythm varies depending on where you are in the gut. In the stomach, it's slow about 3 waves per minute. But down in the duodenum, the first part of the small intestine, it speeds up to about 12 per minute. Then it slows down again in the ileum to 8 or 9.
So the stomach beats 3 times a minute, and the small intestine beats 12 times a minute. It's a huge difference.
Yeah.
But what is causing this beat? The text mentions these interstitial cells of Cajal.
Ah, yes. The interstitial cells of Cajal. They are the electrical pacemakers of the gut.
The pacemakers, so like the ones in the heart.
Very similar in concept, yes. They are these specialized cells that form a network, and they're interlaced with the smooth muscle. They have unique ion channels that periodically open and create these electrical currents. They are the drummers. They don't pull the oars, but they set the beat for the rowers.
That is a great analogy. So the Cajal cells set the rhythm, the slow waves. But if slow waves don't cause the actual contraction, what does?
Right.
We need a spark, right?
We do, and that spark is the second phenomenon, the spike potential.
Okay, spike potentials. These are the true action potentials. This is the real deal.
Yes. Remember, our baseline is madish 56 millivolts. Well, the slow wave pushes the voltage up towards zero, making it less negative. If that voltage crosses a specific threshold, which the text identifies as about negative 40 millivolts, boom, you get a spike potential.
So the slow wave lifts the boat, and if it lifts it high enough over the negative 40 mark, the spike potential happens.
Exactly. And the higher the slow wave peaks above that threshold, the more spikes you get. The frequency can range from 1 to 10 spikes per second. And here's where it gets really interesting for the physiology nerds listening.
Okay.
These spikes are weird. They are very, very different from the action potentials in your nerves.
How so? Because usually we hear action potential and think sodium. Fast, sharp.
In a nerve fiber, yes. An action potential is lightning fast. It's driven by rapid sodium channels opening and closing in a fraction of a millisecond. It's a click. But in the gut uses something different. It uses calcium-sodium channels.
Calcium-sodium. Why does that matter? What's the practical difference?
Two huge reasons. First, these channels are much slower to open and close. This means the action potential lasts a long time. The text says 10 to 40 times longer than a nerve signal.
Wow, 10 to 40 times.
This creates that long, sustained squeeze you need for digestion, rather than a quick, useless twitch. Second, and this is absolutely critical, the entry of calcium is crucial because calcium itself is the actual fuel for the muscle contraction mechanism.
Wait, okay, break that down. So the electricity doesn't just signal the muscle to move. The ions entering the cell actually help physically pull the muscle fibers.
Precisely. In nerves, the electricity is just a signal. It's a message. In the gut, the electricity brings the calcium that causes the mechanical work to happen. It's both the message and the fuel, all in one package.
That connects the physics to the mechanics beautifully. Now, the text also mentions that this baseline voltage, this negative 56, isn't static. It can be tuned, right? Like tuning a radio to be more or less sensitive.
Yes, exactly like that. The baseline voltage can change. If the membrane depolarizes, meaning it becomes more positive, say moving from medic 56 to negative 50, the muscle becomes more excitable. It's closer to that negative 40 threshold.
So it's easier to trigger spikes.
Much easier. Things that do this are, for example, stretching the muscle, the neurotransmitter acetylcholine from the parasympathetic nerves, or specific GI hormones. Basically, when you eat, the system primes itself to fire.
And on the flip side, you can hyperpolarize it, make it more negative, less excitable.
Right. If you drop the voltage down to negative 60 or negative 70, the muscle becomes almost impossible to fire. The slow waves won't even get close to the threshold.
And what does that?
This is what norepinephrine or epinephrine does. This is the fight or flight response. When you are stressed, your body floods with adrenaline, which hyperpolarizes the gut, effectively shutting it down.
We're going to get deeper into that fight or flight mechanism, but before we do, there's one more term in this section I want to define. Tonic contraction. Because everything we just discussed, slow waves, spikes, implies rhythm. Up, down, squeeze, release. But sometimes the gut just clamps down and holds it.
Yes, tonic contraction is continuous. It's not rhythmic. It can last for minutes or even hours. This happens usually because of a continuous barrage of repetitive spikes, or sometimes hormones are causing a continuous entry of calcium that isn't even tied to changes in the membrane potential.
And where would you need that? Why would you need just a constant squeeze?
Sphincters, the valves. Think about the valve at the bottom of your stomach, the pyloric sphincter. It needs to stay closed most of the time to hold food in. If it relied on a rhythm, you'd have reflux every few seconds. So it stays tonically contracted until it gets a signal to relax and let food through.
Got it. Okay, so we have the structure, the syncytium, and we have the power source, the electrical battery. Now we need the control center. Segment 3, the enteric nervous system, the brain in the gut.
This is one of the most fascinating parts of human anatomy, hands down. As we mentioned, this system extends from the esophagus all the way to the anus. It's a complete nervous system in the wall of the gut. And it's arranged in two distinct layers, or plexuses.
Right, the plexuses. If you look at figure 63-4 in the source, it visualizes this network as a mesh. It looks like a net.
A very complex net. And let's break down the two plexuses. The outer one is the myenteric plexus, also known as Auerbach's plexus.
Myenteric. Where does it live and what does it do?
The myenteric plexus is the outer layer, and it's sandwiched right between those longitudinal and circular muscles we talked about at the beginning.
So its location gives us a clue to its function.
It's a huge clue. Because of its location wedged right between the muscles, its primary job is controlling movement or motility.
That makes perfect sense. It's right next to the gear levers.
Exactly. When this plexus is stimulated, it increases the tone of the gut wall, it increases the intensity of the rhythmic contractions, it boosts the rate of the rhythm, and it speeds up the conduction velocity of the electrical waves. It's the accelerator pedal for gut movement.
But the text mentions a crucial nuance here. It's not just about squeezing. It also have inhibitory neurons. That seems counterintuitive. Why would the accelerator pedal also have a brake?
This is vital. You can't just squeeze everything at once or food wouldn't move. It would just be crushed in place. You need to open the doors ahead of the food.
Oh.
So the myenteric plexus secretes an inhibitory transmitter called vasoactive intestinal polypeptide, or VIP. And it uses VIP to relax the sphincters, like the pyloric sphincter leaving the stomach or the ileocecal valve. It inhibits the muscles downstream so the food has somewhere to go.
So it's both the gas and the clutch controlling the flow.
A perfect way to put it. So myenteric is movement. Then we have the inner layer, the submucosal plexus, or Meissner's plexus.
And this one is deeper.
Yes, it's located in the submucosa. Since it's closer to the inner lining, the mucosa, and all the glands and blood vessels there, its job is managing the local environment.
So not the big movements, but the small local stuff.
Exactly. It controls local intestinal secretion, local absorption, and it actually controls that muscularis mucosae we mentioned, the tiny muscles that fold the lining to increase surface area.
So to use your car analogy, Auerbach, the myenteric, drives the train, and Meissner, the submucosal, manages the passengers and the cargo inside the cars.
That's a good way to put it. One manages the pipe, the other manages what's happening inside the pipe. And to do all this, they use a massive chemical toolkit. The text lists over 25 different neurotransmitters.
Yeah, the list is extensive. We know acetylcholine, that's the standard excitatory one. We know norepinephrine, that's the inhibitory one. But then you see dopamine, serotonin, substance P. It really is like a second brain. It uses the same chemicals as the one in our skull.
It creates a rich, complex language. It's not just a binary light switch of on or off. It's slow down a little, secrete more acid, absorb more water, dilate the blood vessels right here. The gut is constantly talking to itself in this very nuanced chemical language.
And while this second brain can run the show on its own, it still takes orders from the head office. That leads us to segment 4, the outside managers. The autonomic control.
Right, the central nervous system's input.
We have the parasympathetic and the sympathetic systems.
Think of these as the hardwired connections between the brain, the central nervous system, and the gut-brain, the enteric nervous system.
Let's start with parasympathetic. This is the famous rest and digest system.
Correct. As the name implies, parasympathetic signals generally increase the activity of the enteric system. It ramps things up. And the text divides this into 2 divisions based on where the nerves come from. The cranial and the sacral.
The cranial division is almost entirely the vagus nerve, right? That's the big one.
Yes. The vagus nerve is the superhighway. It carries signals from the brainstem and innervates, well, almost everything. The esophagus, the stomach, the pancreas, and it goes all the way down through the first half of the large intestine.
So it handles the whole top half of the system.
Pretty much. And then the sacral division handles the back end.
Literally.
Literally. The sacral nerves S2, S3, S4 come from the bottom of the spinal cord and innervate the distal half of the large intestine all the way to the anus. These are especially critical for the defecation reflexes.
Now let's contrast that with the sympathetic system,
Yeah.
the fight or flight.
The total opposite. The sympathetic fibers originate from the spinal cord in the thoracic and lumbar regions, between T5 and L2. They pass through these large ganglia, the celiac and mesenteric ganglia, and then they spread out to the entire gut.
And their message is clear. Stop. Shut it all down.
Yes. The sympathetic system secretes norepinephrine, and it has a very effective 2-pronged attack to shut things down.
Okay, what's the attack?
First, it directly inhibits the smooth muscle, so it can't contract as easily. But second, and maybe more importantly, it strongly inhibits the neurons of the enteric nervous system itself. It basically cuts the power to the local control center.
So it tells the muscles to stop, and it tells the managers of the muscles to stop.
It's a complete shutdown. The text says strong sympathetic stimulation can literally block movement of food.
It can. If you are running from a tiger or, just acutely stressed about a deadline, your body decides that digestion is a luxury it cannot afford.
The factory goes dark.
It shuts the factory down. This is why chronic stress causes so many GI issues. You are constantly keeping the factory in shutdown mode.
You're constantly fighting against its natural rhythm.
There's another component to the nerves that I found fascinating. We always talk about the brain telling the gut what to do, but the text mentions sensory afferent fibers, nerves that talk back.
This is a stat that always surprises people. 80% of nerve fibers in the vagus nerve are afferent.
Wait, say that again? 80%?
80%. That means they're sending signals from the gut to the brain, not the other way around.
So the vagus nerve is mostly a listening device for the brain.
The brain is mostly just monitoring. These nerve endings in the gut wall are sensing irritation, distension, which is stretching, and specific chemicals. If you eat something toxic, the gut tells the brain, and the brain triggers nausea and vomiting. If you are too full, the gut tells the brain to put down the fork. It's a constant feedback loop, and most of the traffic is going uphill.
That's incredible. This communication creates what the text calls reflex loops. That's segment 5. It categorizes these reflexes into 3 distinct levels. I think this is a great way to visualize the hierarchy of control.
It really helps to see the levels of management. Level 1 are reflexes that happen entirely within the enteric system.
So the gut talking to itself.
The gut talking to itself. The brain doesn't even know these are happening. This covers things like local secretion, mixing contractions, local peristaltic effects. It's like your hand pulling away from a hot stove. It's a reflex that happens before your brain consciously registers the pain.
Right, it's all local. Then level 2, gut to the prevertebral ganglia and back to the gut. This is like the gut sending a message to a regional manager.
That's a perfect analogy. These signals leave the gut, go to the sympathetic ganglia nearby, the regional office, and then the command comes back to a different part of the gut. These are long-distance signals within the abdomen.
Give me an example.
The gastrocolic reflex. When your stomach stretches with food, it sends a signal down to the colon to say, hey, new stuff is coming, make room. That triggers a mass movement in the colon. This is why many people feel the urge to use the restroom shortly after a large meal.
Or the enterogastric reflex, where the intestines signal back to the stomach.
Saying, whoa, slow down, we are full down here, stop sending food.
Right. Or the colonoidal reflex, the colon telling the ileum, the end of the small intestine, to stop emptying because the colon is backed up. It's all traffic control.
And then level 3, gut to the spinal cord or brainstem and back. These are the heavy hitters. These are the ones that need the big boss's approval.
These are the reflexes that require central processing. Pain reflexes, for instance, that cause a general inhibition of the whole tract. And of course, the defecation reflexes, which travel from the colon to the spinal cord and back to produce the powerful abdominal contractions needed to push waste out. That requires coordinating muscles outside of the gut.
So we have electricity, we have nerves, we have reflexes. But we are missing the chemistry set. Segment 6, hormonal control. The text provides Table 63-1, which lists the big 5 GI hormones. I want to go through these because they act like the chemical project managers of digestion. Let's do it. These hormones are released from the gut wall into the blood, they circulate through the whole body, and then they come back to hit their targets, which could be in the gut or even elsewhere like the pancreas.
First up on the list, gastrin.
Gastrin is the starter pistol. It's secreted by what are called G cells in the antrum of the stomach. The stimulus is protein in the stomach, distension, stretching, or nerve signals.
And its primary job.
Stimulate gastric acid secretion and mucosal growth. It basically gets the stomach juices flowing to break down the meal you just ate.
Okay, makes sense. Next, cholecystokinin, or CCK. I always struggle pronouncing that one.
TCK, it's a crucial one. It's secreted by eye cells in the duodenum and jejunum, the upper parts of the small intestine. Its trigger is fat and protein.
So when fatty food hits the small intestine,
CCK is released. And it does 2 main things. 1, it contracts the gallbladder to release bile, which acts like detergent to break up the fat. 2, and this is very important, it inhibits stomach contraction.
So it tells the stomach to slow down.
Exactly. It tells the stomach, stop sending food. We are trying to process this fat, and it takes time.
So CCK is the reason why a fatty meal keeps you feeling full for a longer time.
That is exactly the reason. It slams the brakes on stomach emptying. It gives the intestine time to work.
Fascinating. Third on the list, secretin. The text notes this was the very first hormone ever discovered.
It was, back in the early 1900s. Secretin is the firefighter. It comes from S cells in the duodenum. Its trigger is acid.
So when the acidic chyme from the stomach
burns its way into the intestine, secretin is released. It signals the pancreas to dump bicarbonate, which is a base, like baking soda, into the gut to neutralize the acid. It protects the delicate lining of the intestine from being burned.
an essential protective mechanism. Fourth, glucose-dependent insulin-tropic peptide, or GIP.
That's a bit of a mouthful. It used to be called gastric inhibitory peptide, but its main role is a bit different. It comes from K cells in the upper small intestine. It responds to carbohydrates and fats.
That's main job is in the name, I'm guessing. Something to do with insulin.
Exactly. Its main job is to signal the pancreas to release insulin. It's prepping the body. It's an early warning system that says, sugar is incoming, get the insulin ready. It effectively prevents a massive blood sugar spike by getting the insulin flowing before the sugar is fully absorbed into the blood.
That's incredibly smart. Proactive, not reactive. And finally, Motilin.
Motilin is the housekeeper. It's secreted by M cells during fasting when you haven't eaten.
Okay, so this one's different. It's for an empty stomach.
Right. It causes what are called interdigestive myoelectric complexes. Basically, every 90 minutes or so, when the gut is empty, Motilin triggers a powerful wave of contractions that sweep from the stomach all the way down.
What's it sweeping?
Any leftover debris, dead cells, or bacteria, it's cleaning the house between meals.
That's the stomach rumbling you hear when you're hungry.
That is the sound of Motilin doing its job. That's the migrating motor complex.
I love that. Okay, moving on to segment 7. Functional movements. We've talked about the what and the why, but let's talk about the how, the mechanics. The text divides movements into two fundamental types, propulsive and mixing.
Right. Propulsive movement is primarily peristalsis. This is the one most people have heard of. The best visual is to imagine putting your fingers around a tube of toothpaste and sliding them forward. A contractile ring appears around the gut and moves forward, pushing the contents ahead of it.
And what triggers this ring? What starts the wave?
The main stimulus is distension. If you stretch the gut wall at any point, like with a bolus of food, the enteric nervous system reflexively contracts the wall 2 to 3 centimeters behind that point, and that contraction starts the wave moving forward.
The text mentions the law of the gut here. It sounds very official.
It's a fundamental principle. The law of the gut basically says that peristalsis always moves towards the anus. It's a one-way street. But there is a second critical part to it called receptive relaxation.
Receptive relaxation.
For the food to move forward, the segment of the gut ahead of the ring must relax and open up. The enteric nervous system coordinates this beautifully. Squeeze behind, relax ahead. If you don't have that coordination, which happens in some diseases like Hirschsprung's disease, the food effectively hits a wall and can't move.
So it's not just a push, it's a coordinated push and pull. And then we have mixing movements or segmentation.
Segmentation looks very different. If you look at figure 63-5 in the source material, it looks like a chain of sausages. The gut constricts at intervals, but it doesn't sweep forward. It just squeezes, holds for a few seconds, then relaxes. Then new constrictions appear at different points.
It's like chopping vegetables or kneading dough.
Exactly. It chops and shears the food, mashing it against the gut wall and mixing it thoroughly with the digestive enzymes. Without segmentation, digestion would be incredibly inefficient because the enzymes wouldn't be able to reach the center of the food bolus. It's all about maximizing contact.
All right. We are rounding the corner to the final pillar, the plumbing. Segment 8, splanchnic circulation. This isn't just about supplying blood to the tissue to keep it alive. The design here is unique and incredibly important.
It is. The blood flow from the gut, the spleen, and the pancreas doesn't go straight back to the heart like it does from, say, your arm. It all collects into the portal vein, which leads directly to the liver.
So it makes a detour through the liver first. Why?
The liver is the body's main chemical processing plant and security checkpoint. The portal blood carries all the nutrients and also potential toxins or bacteria that were just absorbed from the gut.
So it gets first pass.
The liver gets first pass. The liver cells, the hepatic cells, and the specialized macrophage cells, the reticuloendothelial cells, scour this blood. They remove bacteria and they store or process the nutrients. For example, about half to 2/3 of all absorbed glucose is taken up and stored in the liver right away. It ensures that bad stuff doesn't get into your general circulation. It's the customs agent checking every bag before it enters the country.
The text notes one exception to this: fats.
Right. Most fats are packaged into chylomicrons, which are too big to go into the blood capillaries. They are absorbed into the lymphatic system instead, and they bypass the liver, entering the blood via the thoracic duct up near the neck.
Interesting. Now, zooming in on the blood vessels themselves, figure 63-8 shows the countercurrent mechanism in the villi, those tiny finger-like projections in the intestine. This seems almost like a design flaw that turned into a feature.
It's a fascinating piece of physics. In each villus, the arteriole bringing oxygenated blood up is running right next to the venule taking oxygen-poor blood down. They are in very close proximity.
So what happens?
Because they are so close, oxygen can diffuse right out of the artery and into the vein before it ever reaches the tip of the villus.
So the oxygen short circuits. It never gets to the end of the road.
It does. Up to 80% of the oxygen never makes it to the tip. Under normal conditions, this is fine. The tip gets enough to survive. But in states of circulatory shock, when blood flow is dangerously low, the tip of the villus effectively suffocates. It gets no oxygen. This can lead to ischemic death of the villus tip, causing the gut lining to disintegrate.
That sounds incredibly dangerous.
It is. It's a major vulnerability in conditions like septic shock. But the layout allows for highly efficient absorption of materials from the gut. So it's a trade-off between maximal efficiency and safety under extreme conditions.
Okay, let's talk about how this blood flow is controlled. That's segment 9. Obviously, when we eat, we need more blood in the gut.
A massive amount more. Blood flow can increase up to 8-fold during active digestion. This is driven primarily by local metabolic control.
So the gut controls its own blood supply based on its needs.
For the most part, yes. First, those hormones we talked about, CCK, VIP, gastrin, also act as vasodilators. They widen the blood vessels. Second, the mucosal glands release chemicals called kinins like bradykinin, which are extremely powerful vasodilators.
And the third factor.
The third, and maybe most important, is oxygen demand. As the gut works harder, it uses up oxygen. This low oxygen state, or high metabolic rate, triggers the release of a substance called adenosine. And adenosine is a potent signal for the vessels to open up to bring more oxygen-rich blood.
So the gut creates its own demand. The harder it works, the more blood it calls for. But then we have the nervous control again. The sympathetic system can shut this whole party down.
Oh, absolutely. Sympathetic stimulation causes intense vasoconstriction. It clamps down on the arteries and shuts off the tap. This is a critical survival mechanism.
How so?
If you are bleeding out or running a marathon, your brain and your heart need that blood more than your lunch does. The body shunts blood away from the gut to sustain the critical organs. It can provide an extra 200 to 400 milliliters of blood to the general circulation in an emergency.
But, and this is my favorite term in the whole chapter, there is a fail-safe built in. It's called autoregulatory escape.
This is the gut fighting for its life. It's brilliant. If the sympathetic constriction lasts too long and the gut tissue starts to get damaged from lack of oxygen, the local metabolic factors, like that adenosine we mentioned, build up to such high levels that they override the sympathetic signal.
So the local needs become so great they overpower the central command.
Exactly. They force the vessels back open, at least partially.
Autoregulatory escape. Sounds like an action movie title. But basically, the gut says, I know we are saving the brain, but if I don't get blood now, I'm going to die, and it forces the door open.
That's a perfect description. It preserves the tissue just enough to prevent necrosis. It's this beautiful, delicate biological negotiation between the needs of the whole body and the survival of the individual organ.
So, bringing this all together for the outro, we started this deep dive calling the gut a simple tube, a piece of plumbing.
And we couldn't have been more wrong.
What we've actually described is a masterpiece of engineering. It generates its own electricity with the slow waves and the Cajal cells.
Its own power grid.
It has its own brain with the enteric nervous system making local decisions.
A distributed computer network.
It is a complex chemical management system with hormones that act like project managers. It follows its own laws of physics with the law of the gut.
Right.
And it manages its own blood supply, even knowing when to sacrifice itself for the good of the body and when to rebel with autoregulatory escape to stay alive.
It really is a symphony. And what strikes me is that all of this, the voltage changes, the hormonal signaling, the intricate reflexes, the autoregulatory escape, is happening completely subconsciously. You eat a sandwich, and this massive industrial complex springs into action, coordinating millions of data points, and you don't feel a thing. You just feel fed.
It puts the phrase, trust your gut, in a whole new light. It really does know what it's doing.
It certainly does. And here's a provocative thought to leave you with. Consider the countercurrent mechanism we discussed, where the gut effectively short-circuits its own oxygen supply, making its own tips vulnerable to shock just to be more efficient at absorption.
Right, that trade-off.
And consider the sympathetic shutdown, where the body willingly starves the gut of blood to save the brain and heart in an emergency. The gut is, in many ways, the only organ system designed with a built-in mechanism to be sacrificed for the greater good of the organism.
That's a powerful idea.
Yet it possesses the intelligence, that second brain and the fail-safe, the auto-regulatory escape, to know exactly when to draw the line. It's this constant, high-stakes negotiation happening inside you after every single meal. How much can I give up for the team versus how much do I need to keep for myself just to survive?
That is heavy. And a perfect place to stop. Next time you sit down for lunch, take a second to appreciate the absolute machinery whirring away inside of you. This has been The Deep Dive. Thanks for listening.